Okay, this is the last presentation for the morning, which encompasses all of the health science pitches that we have for UTest 2021. Our afternoon session, which will start at 1 o'clock, is going to be focused more on physical sciences. Each year at UTest, we always have one startup that surprises us with their insight and kind of taking an existing technology and finding a completely different application. From Dr. Keith Party's lab, I would like to introduce you to Glusyn Diagnostics.
Thanks, Hassan. Can everyone see my screen? Let's just confirm that. Okay. So I'd like to start out by thanking all of you in advance for your attention, and also UTest for organizing Pitch Day. So I'm Jennifer, and I'm representing Glusyn Diagnostics, and what we are bringing is a simplified molecular diagnostic workflow for infectious disease diagnosis at the point of care. So as many of you might be familiar, given the ongoing COVID-19 pandemic, there are really two options for diagnostic tests for infectious diseases. That's PCR tests and rapid tests that are available at your local Shoppers Drug Mart or other pharmacies. But the problem is that PCR, generally speaking, is very slow and expensive. It can be up to $200 and several days for patients to receive their results. This creates a barrier to critical care decisions, as well as barriers to access, as samples must be properly collected and shipped by trained personnel. Furthermore, the workflows are too complex to be done at the point of care and are often outsourced to reference laboratories, which employ highly trained personnel and have sophisticated equipment with fluorescent detection and rapid thermal cycling. And unfortunately, the rapid tests are not really a good substitution. Consumer available antigen or antibody tests demonstrate fairly low sensitivity and accuracy compared to PCR tests. They can be useful in pandemic conditions where there's high community spread and you expect to see high viral loads. But under normal conditions, they can be as low as 35 to 50% accurate. So that's where we're trying to kind of bridge that gap using a glucose meter interface for point of care gene circuit-based pathogen detection. So this is essentially an automated workflow based on the integration of a microfluidics device to perform sample preparation, amplification, activation of a gene circuit, which produces glucose in response to the presence of the pathogen, followed by the use of a repurposed off-the-shelf glucose meter that we use to develop a numerical output that can be interpreted as a diagnosis. And in the lab, we just use a conventional glucose meter with test strips. We use a $10 one from Bayer. This technology, the glucose-based detection, is patented. There's a patent filed and held at U of T. And furthermore, recently published data on COVID-19 infection diagnosis has demonstrated substantial equivalence to QRT-PCR on nasopharyngeal swabs for COVID-19. So we chose to focus actually on animal health as our primary target market to go after. So why did we make this decision? So not only can we get the products into the hands of users much faster than we could if we focused on human health diagnostics applications, this is a huge market. It's a $4 billion global market for animal diagnostics with a CAGR of about 8%. Furthermore, about 25% of this market, or $1 billion, is actually focused solely on point of care products specifically. So anything that's not PCR based, that doesn't need to be run in a reference laboratory. And there's growing interest in these point of care products from the veterinary clinician's perspective, with over 65% of sales, even as far back as 2018, being within this category of in-clinic point of care analyzers. And the pet population is growing. 72 million US households have pets as of 2020, growing year over year. About 50% of households in Canada and the US have either a cat or a dog. So our competitive advantage is that we are kind of bridging the gap between these two set tests, as I said before. The Glusyn tests, it can be run for as low as $10 in reagent costs, compared to $100 to $200 for a PCR test. It can also be run in about 2 hours, and this is hands-off time. So the end user would ideally be able to just apply a clinical sample to the device, and it would run automatically, followed by detection on any on-hand paired glucose meter. And the accuracy and sensitivity is comparable to the gold standard of PCR tests. We know what level of sensitivity that we can achieve based on tests in COVID-19 and typhoid. So we are actually, what we're offering is substantial equivalence to PCR tests at a much lower time to result and lower burden to the user. Now, the factors that impact the animal health diagnostics market are some of the same factors that might impact human health diagnostics. There's a few large players and a few small disruptors. The large players, of which I've included IDEXX Laboratories and Zoetis as examples, own about 50% of the market, but they only offer either PCR tests or rapid antigen antibody tests. And in fact, they also pair their PCR tests with the operation of centralized reference laboratories. Smaller players have been really focused mostly on the point of care market, as this is the most fastest growing segment within animal diagnostics. And I'll point out a couple of examples. Advanced Animal Diagnostics is focused on agriculture, point of care hematology analyzers, and they raised about $50 million in total funding in recent years. Furthermore, Zoetis actually in 2018 acquired a smaller company called Abaxis, specifically in order to onboard their point of care products. And they acquired them for about $2 billion, in spite of the fact that their point of care products are these rapid antigen antibody tests, just as an alternative to PCR to their customers. What we offer that none of these companies offers is that Glusyn Diagnostics offers actually a brand new way to do point of care tests, where we get the accuracy and sensitivity of PCR with the ease of use of rapid antigen tests. And our revenue model is going to be very familiar to you as kind of the razor blades model. So what we'd have as our end users is veterinary clinic staff, and this can be anyone who can read an instruction booklet, from the receptionist to the veterinary clinician themselves. And the sustained sale of single-use cartridges with freeze-dried shelf-stable reagents, which have been demonstrated to be useful for cell-free systems. Furthermore, because of an established pipeline for the design of the tests for different, all sorts of different types of pathogens and potentially different sample types, there are going to be opportunities for licensing the technology for wider distribution once we've got our proof of concept. So the team are all members of Dr. Keith Pardee's lab and the Faculty of Pharmacy at University of Toronto. And Dr. Pardee is not only an accomplished academic, he's also co-founded several companies coming out of the lab, including, for example, Life Science Key Technologies, which is a Y Combinator-backed company operating out of Velocity in Waterloo, and Liberal Biotech. Evan Amalfitano is a founding member of the Pardee Lab, having joined in 2016 as a PhD student. And he's actually the inventor of the technology. And he came up with this idea because he wanted to build an electrochemical sensor using the established cell-free systems that Keith Pardee developed in his postdoc. But he didn't really want to build something new. So he had this idea of repurposing one of those widely available, accessible, and cheap electrochemical sensors in the world, the glucose meter. Myself, I'm Jennifer. I joined in 2020 to work on some pandemic response projects. And I actually started my career out in industry, working on in vitro diagnostics, including assay development, verification, validation, and clinical trials for 510(k) FDA pre-submissions. Moise Charania is a research associate and lab manager here in the Pardee Lab, and he also joined to work on this project in 2021. He has an impressive list of publications and citations and has experience working in labs in in universities and hospitals, national labs in the US and Canada. So for the technology development, this has been a long time coming. Even from far back as Dr. Keith Pardee's postdoc. And this has really accelerated in the wake of the current ongoing pandemic. So in October of 2020, the Party Lab was awarded a huge grant for pandemic response in order to set up this collaboration with the NRC for microfluidics automation of the tests. In February of 2021, Evan's work was finally published in Nature Communications, where they were able to demonstrate that this technology can work for the detection of typhoid, as well as fluoroquinolone resistance and COVID-19. And just recently, we were awarded a $20,000 NSERC Ideas to Innovation grant to do a market assessment specifically for the animal health market applications of this technology. So our focus and our funding roadmap closely aligns with our R&D roadmap. So in the winter, we're going to be doing this market assessment. We've contracted a firm out in Guelph that has specialization and expertise in animal health and diagnostics. And what we're looking to do once we go through the market assessment and identify which disease targets we want to focus on, we're going to shift gears to product development. So what we're looking for in 2022 is to secure a funding partner for this development. So we're going to be looking at $125,000 in the first year, potentially through a phase 1 NSERC Ideas to Innovation grant. Following development of our first target that we want to go after, we're going to be looking for seed partners or funding for 2023 to actually do manufacturing process development and limited clinical field trials in order to go to market. At which point we'll be looking for up to $400,000 to maximize government matching. So thanks again for your attention. Please feel free to contact me through my email listed on the bottom, or come visit our booth in the session room. Thanks.
So I do have to make the comment. It's genius that Evan found the path of least resistance. I always say it's the laziest path that can get you the most innovative solution. So kudos on that. First question: could you speak about the regulatory path in the animal health market? Do you need to show equivalency to PCR results for each infectious disease?
So that's a really great question. So the animal health diagnostics market is regulated in Canada under the Canadian Veterinary Biologics framework. And there are different regulations depending on the type of target that you're going after. So there's reportable diseases like African swine fever that the government is very interested in tracking. Those definitely are kind of very focused and require this centralized testing model. And then there's diagnosis, which does require it requires less clinical evidence than for human health diagnostics, but there is still you're going to have to do your verification, validation, clinical trial, and limited clinical trials. Although there is an exception for research use, which can actually get us a little bit further ahead. And then, if there's testing for the agriculture sector, there's actually really interesting market for testing herd level. So not doing individualized diagnosis, but actually testing samples within that environment. And that is completely unregulated. So there's definitely different opportunities that we've identified within that framework.
Okay, next question. Are you aiming to license the technology rather than exiting through merger and acquisition?
So we're kind of open to different, there's different ideas. So we're very early days. So we founded just to join UTest, so just in June of this year. We're not yet incorporated, so we're still working on developing our business model. But we see that those are definitely two paths that we could go down, for sure.
Okay. Do you plan to make your own instrument at some point?
So we're collaborating with the NRC to develop the microfluidics device. So this has been a project that started last year and has made very, very good progress. We have our prototype that's being tested right now. But the device is actually going to be fairly simple to build and manufacture. So we've thought about that from the very beginning. The cartridges are injection moldable, so we know that they're going to be manufacturable at a really low cost, probably pennies per unit. And the device is designed to run isothermal assays, so it's not really much more complex than a hot plate, which is great.
Okay. Who is your ideal customer, and who is the end user, and who is the buyer of your portfolio? Those three questions.
3 questions, yeah. So the end user is again going to be, so we're focused to start with on the veterinary clinic. These users would probably presumably have glucose meters as well on hand available to pair with the device. Our customer, we'd probably need to figure out distribution channels, which we're kind of in the process of as, the animal health diagnostics market. There's different ways that customers are going to be buying our products, different distributors that we're going to be looking to partner with, as well as reagent suppliers.
Okay, great. Looks like we have ended with the last question. Thank you very much, Jennifer, for the presentation.
Thanks, Hassan.